Hello, welcome to Biology 23, Introduction to Infectious Disease. My name is Cheryl Hackworth, and I will be your instructor this semester. Remember that in addition to watching these videos online, you should also be reading the chapter every week. The chapters from the textbook will sometimes cover material that is not covered in this lecture video. And some information covered in the video lectures are not covered in the textbook. You do need to be watching both, and you do need to be taking notes on both the video lecture as well as the textbook reading. Overlapping material, and by that what I mean is material that's covered in both the textbook and this video lecture, is most likely going to be information that will be on your quizzes. However, textbook questions or homework assignments could just be from the textbook or other information found online. But because you'll be asked as part of an assignment to take notes and submit the notes to this online video, you should have a Word document open or a piece of paper and pencil ready to take notes on this lecture that you're about to watch. All right then, let's begin. Chapter 1 begins with a discussion about the thoughts of where life originally comes from and the philosophies that occurred around this idea for thousands of years of human existence. Prior to invention of the scientific method, there were two main ideas of how life arose. One of these main ideas was spontaneous generation. Spontaneous generation is the idea that life comes from non-life. So for instance, if you leave a piece of meat or dead food, like a piece of bread, out on a tabletop, you may come back a day or two later and discover that there are maggots or insects crawling around on that food. Early humans thought that it was possible that the maggots actually arose from the dead or non-living bread piece or piece of meat. Of course, nowadays we know that this is a result of flies laying eggs on this food. But the idea of spontaneous generation was widely accepted by many people for thousands of years. However, there was a competing idea at the time, and this is the dominant idea today, and that is that life cannot arise from non-living things, that life can only arise from living things, and so if you see a living thing, that has to be offspring of another living thing. That is referred to as biogenesis. Spontaneous generation, therefore, is sometimes referred to as abiogenesis. Now, what we're going to cover in this chapter is going to be some information that will seem fairly antiquated to you. But in fact, these were the common philosophies or ideas prior to the use of the scientific method. The first thing that we want to cover in this chapter is the first ability for humans to visualize or see living systems that would transmit infectious disease. So how did we first see these infectious agents, what we now refer to as microbes? Well, the first individual that really saw this was Robert Hooke. Robert Hooke lived from 1635 to 1703, so you could essentially think the late 1600s. He was credited with coining the term cell because he was the first individual to invent the compound microscope. The compound microscope was the ability to take two lenses, like two magnifying lenses, and put them together in order to see small things. When he did this, he was able to take a look at smaller structures in things like tree bark, bones, insects, plants, and mold. And you can see some of his drawings here. These structures were only really at a magnification of 35x, which was phenomenal at the time, but compared to today's 1,000 or 2,000x with light microscopes, is orders of magnitude less. Robert Hooke's concept of small living things, small structures invisible to the naked eye, was expounded upon by Anton van Leeuwenhoek, sometimes referred to as Antony van Leeuwenhoek. Anton van Leeuwenhoek lived almost exactly at the same time as Robert Hooke, and had in fact read some of his papers and learned a lot about this small world. Anton van Leeuwenhoek, due to his very deliberate and careful ability to generate small lenses, was able to in fact build simple microscopes, so they only had a single lens, but with much more powerful magnification. His magnifications were in fact almost 10 times stronger than Robert Hooke's. And with this better magnification, he was able to see lots of more complex things. He took a look at a lot of freshwater samples, teeth scrapings, which would of course include bacteria from your teeth and saliva, soil and rainwater and well water and seawater. He also took a look at mold and algae, much like Robert Hooke did, but also took a look at small little animals, frog eggs, and even the sperm of many different organisms. From this, he was able to generate drawings. And in his simple microscopes, he was able to watch as some of these little organisms seemed to move around. He referred to these little organisms as animalcules because he thought they were small little animals. Nowadays, of course, we refer to these as microbes. Now, after that, people sort of recognized that there were small living things that were invisible to the naked eye, but could be made visible by using light lenses. This study was interesting, but didn't really result in any additional knowledge or information, especially as it pertained to human health, for hundreds of years. This is why, during the Crimean War, when the Russian military invaded Turkey, that we saw some of the first advancements in human health. This is in the mid-1800s, so a good 200 years or so after Robert Hooke and Anton van Leeuwenhoek's inventions and discoveries. During the Crimean War, a very famous nurse, Florence Nightingale, was asked by the head of Britain's War Office to be superintendent of all female nurses and train the female nurses in the British military hospitals in Turkey. Florence Nightingale, because she was a female, was not treated as equals among the military generals and admirals. But she worked very diligently in hospitals and had an interesting philosophy at the time because she believed in the quality of sanitation. She was referred to as a sanitarian. Sanitarians who believe in sanitation emphasized and promoted adequate ventilation and cleanliness in areas where there were wounded and ill soldiers. She insisted that windows and fireplaces be left open in order to allow fresh air to freely circulate in wards. She also encouraged proper nutrition, pure air, clean water, efficient draining of waste from facilities, and general cleanliness, which included hand washing and bandage dressing changes. She also insisted on a quiet, peaceful atmosphere to help with the mental health of her patients as well. Her tireless efforts ended up resulting in many soldiers' lives being saved, and publications of hers ended up being sold worldwide. Another heroic female nurse at the time, Mary Jane Seacole, who applied to work for Florence Nightingale but was turned down, was one of only 3 Black nurses that worked at the time of the Crimean War. While she couldn't work for Florence Nightingale, she ended up raising funds and setting up a hotel, store, and restaurant where she could service ill and sick soldiers as well. She was able to apply information and knowledge that she had about herbal remedies from the Caribbean to treat soldiers that were suffering from diarrhea, dysentery, and other illnesses. She was again lauded by the soldiers that she served. These efforts, though resulting in publications that were widely read, didn't end up significantly altering the way ill or sick people were treated for quite some time. In addition, all of this knowledge and information was being used by the British military. And the Americans, quite some distance away, were not able to gain the benefits of such knowledge as easily. This is unfortunate because during the American Civil War, far more men died from infection than from gunshot wounds on the battlefield. Infection, in fact, was a very deadly issue during the American Civil War and cost many, many lives. Partly, this was because when soldiers were wounded on the battlefield, their infections led to death. But in addition, any soldiers that were rescued from the battlefield wounded and brought to surgeons, the surgeons would end up resulting in infections as well. Everything about the surgeries during the American Civil War was what are referred to as septic. Septic is a way that we describe today the ability to transfer microbes or containing microbes. So many of the procedures in surgeries during the American Civil War transmitted microbes and transmitted infections, resulting in numerous deaths. Often, knives were not cleaned properly. Bandages were reused, transmitting infections that way. And no one really quite understood exactly how transmission of these diseases were caused, where these infections came from, or how to stop them. In fact, most people at the time considered that the air was responsible for transmitting infections. These were referred to as miasmas. It was the bad air and not the surgeons and their filthy hands or filthy tools that were continually blamed for transmission of infections. There were some surgeons at the time that noticed that if they used some chemicals on the bandages or on their hands, that they would end up reducing the number of dead or dying in their hospitals. This included chemicals like bromine. Bromine is a chemical that's similar to chlorine, which is what we use in bleach or to disinfect water. And bromine is fairly effective in killing microbes. By soaking bandages in bromine and using these bromine-soaked bandages to treat wounds, the surgeons were in fact using a very early form of antiseptics. In addition, boiling bandages or sponges or sutures was another means of reducing the number of microbes, though again, no surgeon at the time knew that this was what was happening. They just associated the use of boiling and some chemicals with the reduction in deaths. In the next section, we'll be talking a little bit more about transmission of disease in hospitals and some of the discoveries associated with reducing this disease transmission.